Okay, so yeah, thanks for coming, everyone. And some of you who are familiar with my work, that there's going to be a bit of repeat here, but there should be some new material as well. So yeah, I want to talk about reporting bias, also called publication bias. There's a lack of consensus on the terminology, to be honest. And I think one thing that needs to be done in the field is to sit around and just hash out what exactly we're going to call these things. So some disclosures to begin with. As Aaron said, I was a reviewer at the FDA from for a 3-year period from 1998 through 2001. And then after coming to Oregon, leaving the FDA and coming to Oregon, I did some consulting for a couple of years and some promotional speaking. But then I soon realized that wasn't for me. And so I like to say that I've been in recovery since 2005. So anyway, what is reporting bias? It's by definition, it's when the publication of research results depends upon their strength and their direction. And we'll talk about this next bullet a bit more later. Who's responsible? It can be when these negative or inconclusive results are less likely to be either submitted by authors or accepted by editors and reviewers. So it's a tango. It takes two to tango, as they say. No exception here. And it can apply to entire trials, which I call it study publication bias because it makes it pretty clear that you're talking about an entire study. Or it can affect the outcomes within those trials. And that's also goes by many names: outcome reporting bias, also called outcome switching, for p-hacking, harking, which stands for hypothesizing after the results are known. I love that one. So the consequences of reporting bias: it affects both efficacy and safety with the on the efficacy side of things. And by the way, the efficacy safety dichotomy translates into the clinical world. We think of the risk benefit ratio, of course, clinically. So on the efficacy side of things, there's sort of like a great inflation, if you will, where everything gets an A. And if everything gets an A, then you don't, and no one gets an F or a D or whatever your grading system is in your country. If no one fails, if no one is mediocre, if everyone, it becomes like Lewis Carroll's Dodo Bird, who said, everyone has won and all must have prizes. Well, if everyone's, if all you see are positive studies, then you won't be able to distinguish, good drugs from bad drugs. It can affect safety as well. Harms can be concealed or played down. And so overall, when you take these two things into consideration, it will wind up skewing the risk-benefit ratio. And on the clinician end of things, it'll create an overenthusiasm about treatment options. And scientifically, we always hear about evidence-based medicine. Well, it's also, it's been said that it's actually evidence-biased medicine. So here's a recap of our study that we did, and we published in 2008 in New England Journal of Medicine. And so some of you, perhaps many of you, will have heard this before, but so forgive the recap. But so basically, what we did, with the study, we identified a retrospective cohort on 12 antidepressants, and all the studies, all the clinical trials that had been registered with the FDA. And I want to emphasize the word registered because that's a key strength, I believe, of using FDA data, is because the FDA finds out about studies before they are conducted. And that's key, because before a study is conducted, everyone's optimistic. The drug companies believe, of course, all our studies are going to be positive. It's only later on that oops, it didn't quite turn out that way. Well, they can't change their tune later and not submit a study report to the FDA, because the FDA will know, well, what are you trying to pull here? We know that you're going to do this study. We know you did it. Cough up the data. So we had the data on all the studies. We knew about all the studies or had been registered with the FDA. And then using the FDA review documents, which are available online, by the way, we use that to identify all those studies, regardless of their outcome. And then we could track each study into publication, first of all, asking, was it published at all? And secondly, if it was published, was it spun, or was it published in a way that agreed with the FDA's conclusion about the study? And if it did deviate from the pre-specified primary outcome, then basically it amounts to, if you, I guess, maybe this isn't so relevant now, at least in the US, that since the elections were a couple of months ago, but it's basically like a broken campaign promise. Someone, politician says that they're going to do something, and then or a breach of contract. What's put in the protocol, the primary outcome, is like a contract with the scientific public, is that this is what we believe is what the study is going to live or die on. But unfortunately, it doesn't necessarily get honored later on using the classic peer review system. So here is this is a figure that was not in the New England Journal, but this is sort of a summary of it. This is kind of how it looks if you are, if based upon one's reading of the journal articles. And basically everything was positive, and by positive, I mean, it's everything above that red line was statistically significant, appeared to be statistically significant on the primary outcome. As you see, only a few studies, very few studies, were acknowledged as being not positive in the journal articles. And those are, of course, the 12 antidepressants that we looked at across the bottom there. But then, if you looked at the FDA's version of the same trials, you get a very different picture, and you see that, lo and behold, there are a lot of negative studies. And basically, it was about 50%. So 50% were positive, 50% were not positive. Quite a difference. So it's I have a GIF on my in my Twitter account where it toggles back and forth between these two. So I'm going to toggle back and sort of now you see it, now you don't. So those negative studies, there they are, and they're gone again. And so it's the journal articles are not not accurately representing what the truth is. So what became of all those negative studies? Well, they were, I mean, did they, you had a lot of them that were unpublished. You had 22 of them. 74 was the total number of studies that we looked at, and 22 of them were unpublished. And then there were there's another 11 of them that had been spun from negative to positive. And there were just, a tiny number that were where they admitted to a negative finding. So these are the 11 studies, and you may have heard the expression putting lipstick on a pig, trying to make something look positive or look good that's really kind of ugly. I don't know, maybe pigs aren't so ugly after all. But so there's a number of studies here, and you can see that they were like the first line, for instance, here's Acta Psychiatrica Scandinavica in 1992. According to the journal, a p-value was less than 0.05, but you look at the FDA review of the very same study, and the outcome, the true result on the primary outcome was 0.316. And a number of other studies like that. So how did this happen? Are these just blatant lies? Do they just make it up out of thin air? Well, what they did is, and I think going with the putting lipstick on a pig, let's talk about colors of lipstick. And they are different analytic methods that were done post hoc that how they conflicted with the FDA's way of doing, the way that they had agreed to do in the protocol, and kind of what they did after the fact. Here are some of the enhancements, if you will. So the 6 cases, the most common maneuver, if you will, was how dropouts were handled. There's always going to be dropouts in clinical trials. And statistically, there need to be ways of accounting for them, because you do want to account for, you don't want to just ignore the people that dropped out, because usually there's the intent to treat principle. People that go into the trial represent a random, hopefully, well, as close as you can get to a random sample of the people with the disorder in question, in this case, major depressive disorder. If you look only at the people who make it to the end of the trial after people have dropped out, you're now looking then at a rarefied subset of patients, people who have tolerated the drug particularly well from a safety standpoint, or perhaps done better from an efficacy standpoint. So they're no longer that intended random sample or intent to treat. So anyway, the dropout, just ignoring the dropouts and doing an analysis on the people that made it to the end of the trial, a so-called observed cases approach or complete cases approach, was probably the most common way. And that way they could get different results and get a statistically significant p-value, when in fact the intent to treat approach was not significant. The second one, efficacy subset, is a variation on that, where they made up some rules after the fact. Oh, we'll count them if they make it to, through a certain number of visits, but not after that. There was another case of, or 2 cases of, where you had multi-center studies, and they cherry-picked out a single site that happened to do well, and published on that, and didn't publish it as a multi-center study, which was, of course, how it was intended. And then another one, or an inclusion of a site that the FDA had disallowed because there were some shenanigans, I believe, going on with the blind, that they thought that the blind had been broken. I think that was the issue. So we talked about studies being positive or negative, and that's probably, from a statistical standpoint, that's an oversimplification, because it hinges on a p-value of being less than 0.05 or greater than 0.05, and don't want to get too much into the weeds on this, but it's just suffice it to say is oversimplified to because if it could be that 1 or 2 patients can make the difference between toggling on one side or the other of 0.05. A more statistically rigorous way of handling these things is through meta-analysis, where you use all the data, and you were able to combine data from different studies and combine into a single bottom line for each drug. And in this case, so we looked at that. I think you can see my cursor. The squares here are the FDA-based effect size, and the open circles are the journal-based effect size. And here are the 12 drugs, and here is, as you can see, there's a dark line connecting the FDA-based effect size to the journal-based effect size, and in every case, for each of the 12 drugs, the line slants down and off to the right, consistent with an increase in the effect size. So if you put them all together, if you put all the 12 antidepressants together, you get a 0.31 standardized mean difference, which we probably don't have time to go into today, versus a 0.41 for journal-based. So it's an exaggeration of the efficacy from 0.3 to 0.4 roughly, or about a 32%, so roughly a third boost in apparent efficacy. Doesn't look as dramatic as it did with the toggling, positive versus negative studies, but the underlying message is still the same. What's happening is that the effect size has been inflated, and the reality is that the drugs are less effective than has been conveyed by the published literature. And just a parenthetical point, I know sometimes there's a lot of discussion about, some people, bashing on antidepressants as being ineffective. Less effective is not necessarily ineffective. It's something can be, it's like, now I'm getting thirsty here as I talk. There's the old glass half full, glass half empty thing. The glass can be, you drink it down, it doesn't, if it's not as full as we would like it to be, it doesn't mean it's completely empty. That's a little bit of a side point, but perhaps we'll have time to talk about it later. But it's not just antidepressants. We've, with some colleagues, I've looked at other drug classes, drugs for anxiety disorders. Actually, these are the second generation antidepressants. They're actually the same drugs, but for a different indication, in this case, for 5 different anxiety disorders. And we found, again, an exaggeration of efficacy due to publication bias or reporting bias. Also looked at antipsychotic trials, basically the same story, not quite as bad as with the antidepressants. And then this third one down here is not something that I was involved in, but I just love the title. Why olanzapine beats risperidone, risperidone beats quetiapine, and quetiapine beats olanzapine. So A is better than B, B is better than C, but somehow C is better than A. How can that be? So what they did is looked at just an exploratory analysis of head-to-head comparisons of the second generation antipsychotics, and basically it comes down to picking and choosing as to which trials are going to get published or not. So imagine the drug company they might do a number of trials pitting their drug up against a competitor, and imagine if the home team might win, and if the home team wins, then everybody gets to know about it. If the home team loses, well, we'll just sort of keep that a little quiet there. Now, it's not just drugs, though. We've got psychotherapy for depression. This is a study that I was involved in together with actually all my colleagues in this were psychologists, to their credit. So they were, and to their credit, because they were willing to take a critical look at their own discipline. And what we did is we used what's called an inception cohort, trying to identify trials at their inception, which is basically what we had done with the antidepressants. And by having the access to the FDA data, we knew had information about studies from the time of inception. So in this case, with psychology studies, psychotherapy studies, there is no FDA, of course, but there are grants. And so we used an NIH grants database called Reporter to identify all the studies that had been done. And then we were able to track those into publication. And we found that about 3/4 of them, this blue portion here, were indeed published, but about 1/4 were not published. Now, what we don't know about these blue ones here is, as I have here, unable to rule out outcome reporting bias. So we were unfortunately left to take their word for it that there wasn't any outcome switching or p-hacking, which seems rather, I guess we'd have to be naive to believe that every one of these trials was truly positive. There's probably some p-hacking going in there as well. So this is overly optimistic, but suffice it to say that there is publication bias in psychotherapy studies as well. And if you then, we did a meta-analyses based upon, with versus without these unpublished studies. And we found out the bar on the left is another presentation of what we had found with the antidepressants in the New England Journal paper, an exaggeration of 0.31 to 0.41. Well, with the therapy, there was a bump up or a boost in the standardized mean difference from 0.26 to 0.37, and just coincidentally, almost identical. The increment was almost identical to the one for the medications. Here's a title that I think just makes the point. There's this false positive psychology, undisclosed flexibility in data collection and analysis allows the presentation of anything as significant. So what about other medical fields? Well, here's a narrative review article published in Trials in 2010, and they looked at 40 indications with comprising about 50 different pharmacological, surgical, diagnostic, and preventative interventions, and they found evidence for publication bias in areas such as pain, migraine, cardiovascular disease, gastric ulcers, irritable bowel syndrome, and on, hypercholesterolemia, and so on. So the psychiatry has company, and apparently a lot of company. So how is this possible? Why is this happening? So is the hallowed peer review system not everything that we've been told? And so why do we have these, these discrepancies between, in the case of the antidepressant study, FDA and the journal articles? Well, if something is, the problem is with the peer review system, there are some loopholes, which talk about in a minute, and the way it's conducted. The FDA review closes many of those loopholes. And those loopholes are also closed through a system of peer-reviewed publication called register reports, and I'm going to touch on that towards the end. So here's basically the how information flows in the peer review system. Someone conceives of a study, they write their protocol. Protocols often only for the eyes of perhaps the grant review committee and the investigators themselves, that you might plan for a number of scales. If you're looking at depression, for instance, you could do the Hamilton Depression Rating Scale, you do the MADRS scale. You might as well. You've taken all the trouble to recruit these people. You've hired study coordinators and raters and so forth. You probably want to do the Clinical Global Impression Scale, and there's 2 flavors of that. And you can throw in some self-report scales that you hand to the patient, like the PHQ-9, and so on and on. So you get a whole bunch of, you have a lot of data that you can collect, different looks at the same underlying phenomenon. But one of them should be the primary. It needs to be, you have to pick one as being, that's the hypothesis. Usually you don't say something is going to turn up positive. I think something, you can't, because the problem is one of multiplicity, and as I have there in parentheses, shotguns seldom miss. If you spray a target with a shotgun, you're gonna hit something. You're going to get lucky and hit the target, but that's why you have to guard against multiplicity and pick a primary up front. Anyway, you go on and you get your funding, hopefully, and you get the regulatory approval, hopefully, and then collect then the data is collected, then it's analyzed, but then, oops, no, you don't have that P less than 0.05. Well, then what happens is you get to try out alternate methods as needed. And as the saying goes, if you torture the data long enough, it will confess to anything. It would have to be a pretty bad data set. If you can't torture the data if it doesn't confess to a P less than 0.05 with something, then you really got a super negative study. Usually something put enough lipstick on that pig, eventually something will turn up. So then and only then, after all this, years have gone by since this study had been conceived of, then you write it up or not. So if you choose to, you simply not publish it. When you choose to, you can delay it for a while. And how you choose to, outcome reporting bias, and the system invites harking. Again, harking is this hypothesizing after the results are known. So you can, again, decide once you've been enlightened by the data, which methods you used were flawed or which ones were valid, and which ones may or may not merit emphasis in the manuscript. So it's like picking. It's like you're at a horse race, and rather than picking a horse and having to live or die by that, you get to wait to see which horse wins the race and then place your bet. Well, that's totally absurd, but that's exactly what happens with, or what has been happening in the world of peer-reviewed publication. You get to see what turns out positive, and that's what you get to emphasize. And then the reader on the other end doesn't get to see, that doesn't know about all the stuff that went on in the background. So a journal probably doesn't have a way of checking that unless they get the original protocol. And so it very much it's handled by the honor system. Now, there's an exception to this. Journals could know. Again, they could ask for the original protocols, and there are a few journals that have recently started asking for that. An even simpler way is to check clinicaltrials.gov and see at the registration step. Now, this is something that's been around only, relatively recently. Well, it depends on your time frame. Recent to someone old like me, but it's ancient history to someone who's younger, of course. But anyway, people could be checking clinicaltrials.gov and comparing that with the manuscripts, but unfortunately, that's not being done very much. Anyway, after all this happens, then the submission step comes finally. And then final steps, the journal will do its thing. It can reject, or the journal might engage in some harking as well. They might say, I think you should try this other method. And then finally, the publication, and then there's the news coverage, and the reader again on the the final end user of the information believes that, well, it's in peer review, it must be true, and the reader believes that, it assumes that the published study is representative of what happens when the drug A is used in condition B. And the reader will also assume that whatever's emphasized in the title or in the results section is consistent with the original hypothesis. But that may be, and it may not be. So who's responsible for this? You have industry versus academia. Well, of course, the pharmaceutical companies are, they are corporations, duh. Their mission is to serve their shareholders, and they can best do that by increasing drug sales. And how are they going to increase drug sales if they say, yeah, our drug that we want to sell you doesn't work? That's not gonna sell the drug very well. And then they have their key opinion leaders, or KOLs, also called thought leaders, going around giving talks, and they get paid big bucks to go around the country and talk it up at meetings. This, in my field, the American Psychiatric Association, and cancer, ASCO, and so on. And they are often under confidentiality agreements, so where they may not be able to divulge the negative results. But there's also evidence of publication bias in academia as well. And some people have done studies looking at government-sponsored research. Kay Dickerson did a very study quite some time ago, in 93. My gosh, how many decades ago is that now? So she's, and she just retired recently, but she was a real a beacon in this area. She led the way for most of the rest of us. More recently, but still a long time ago, a couple of decades now, An-Wen Chan used data from the Canadian Institutes of Health Research, basically since you guys are largely Canadian, you know it well. And he was sort of an insider at CIHR, and he was able to get the protocols for a cohort of clinical trials, and he was able to compare them with the publications, and he found discrepancies, shall we say, that there was evidence for outcome switching and picking and choosing as to what was emphasized and what wasn't. And so in academia, you've got these incentives. You've got pilot data must show promise. Otherwise, your tenure may hinge upon that. Do people like having a reputation as being successful? I've got King Midas there. That whatever they touch just turns to gold. P less than 0.05. Psychological things such as confirmation bias. People like to be right. They don't want to be known as someone who spent years working on a certain area and then, hey, it didn't work. They want to be, thought of or think of themselves as doing something that worked or would help, might actually turn out to help patients. There's the phenomenon of hindsight bias. Looking back, I knew it all along. Like, picking your outcomes after the fact. You can easily convince yourself that something that this was the right way of doing it. The way I was originally thinking about that was actually that was flawed. Social proof is kind of looking at your peers and mentors and saying, well, they do it this way, and this is the way it's always been done. So I guess that's the way it should be done. That's the way the game is played. Not thinking that it's, maybe this isn't right. Journals like marketable findings, they've got their incentives as well. Impact factors such as big name journals such as The Lancet. If you look at their instructions to authors, it says they prioritize reports of original research that are likely to change clinical practice. And systematic reviews should have a major effect on human health. So if you have a negative study, that's basically telling you, forget about it. They don't want to see it. It's not important. And it's not going to help their impact factor because, they're going to get more eyeballs if they publish something positive, and the news is going to cover it and, get more eyeballs and subscription. It helps with subscriptions. Now, journal reviewers are human too. The process is they also get biased by whether results are significant. And I'm going to have to speed along because my time is starting to run short. But anyway, there are some promising new developments. This is a graphic of what's occurred over the last couple of decades here. The creation of clinicaltrials.gov, and here's a lawsuit was part of what that helped kick things into gear as well. A number of developments here, which I won't go through this one by one, but you have the registration of studies before they're done. And more recently, in September 2008, there was the first requirement that they should report basic results. Well, that wasn't actually quite done yet. That's been reinforced more recently in a final rule, they called it. But and there's not 100% compliance with that either. But suffice to say, there's been a lot of activity. There's also been a lot of papers, such as papers that I've written, but a number of it. I'm just one of many people in this area that are paying attention to this, and that's putting pressure on drug companies, among others. So you can use clinicaltrials.gov, and you can use it to see if all studies are published. And here's a study that I found out about at through an FDA advisory committee meeting back in February of 2019. And this is a study of esketamine, or brand name Spravato, and as you can see, the p-value is 0.06. So it didn't make it below the magic threshold of 0.05. In fact, they seemed to get lucky here between day 22 and day 28, week 3 and week 4. It almost made it, but anyway, they tracked, drug and placebo were basically indistinguishable for most of the study. Anyway, it didn't. Obviously, it was a negative study, and to find but there are ways of tracking these things down now and seeing if they've been published, and if they've been published in a way that's truthful, which we didn't used to have these at all. We used to be totally at their mercy. They could publish or not publish with impunity. So going into clinicaltrials.gov, you can search for esketamine, and here is a case where they actually were compliant with posting the results in clinicaltrials.gov. I won't show you those results, but I want to show about the publication. Well, okay, so here in June 2020, this is how it looked. And in clinicaltrials.gov, there was no publication. But here now, you can see that there are some publications. But are they really? There's publishing and there's publishing. So it turns out that this was published, but it wasn't published as a standalone negative study. When a study is positive, then it gets published. Tell the world we have a positive study, or a whole article devoted to that. But if it's negative, well, this is what happens a lot. You wind up with a review article that includes the study in question. So they talk about data from three induction studies and 1 maintenance study. Anyway, it's buried in there, and so it doesn't make the same impression upon the reader that, oh, there was a negative study there. It's sort of wrapped or bundled in this package, makes it smell better. So this brings up another type of reporting bias, a pooled reporting bias, I guess it's called. So this is a paper that I was a co-author on, and it was led by some folks, colleagues of mine in Holland at University of Groningen, Imke Anna de Vries, and hiding negative trials by pooling them. So I just want a quick reprise of this previous slide about the fact that it's after the fact. You can write it up or not if you choose to, when you choose to, and how you choose to. So how do we, is there a way around this? And I touched on register reports earlier. And this is where you actually have, and I think this is an actual, I'm excited about it. I talked about inception cohort previously, about the how FDA reviews are useful because they know about the studies before they're done. And again, before the study is done, everyone's optimistic. It's only after the fact, when some turn up negative, that people start hemming and hawing and say, well, I should try this other way, or maybe we don't publish it. So with register reports, there is a stage 1 peer review, which is before the data have been collected and analyzed. And then stage 2 is basically what we have now after everything is done. So stage 1, I think this is at the time the study is designed, and I think it makes sense to me, at least, that you should be able to judge whether the science is good based upon the methods and the rationale for the study. You don't need to look at the results and find out whether it turned out to be statistically significant, because then you've got the hindsight bias. Oh, well, and as I think about it, maybe that wasn't such good science after all. And that's what register reports, this approach addresses, because there has, in their doing this, they found that there have been reviewers that get to stage 2 and they start saying, maybe this non-significant study, maybe we made a mistake, and they've told, no, you can't do that. You already said they have to make a preliminary publication decision at stage 1. Now they're not locked into it entirely, because maybe they don't recruit, maybe they only recruit 10% of the number of patients that they promised to report or something like that. But anyway, the principle remains. So there are 300, over 300 participating journals as of now. The emphasis has been, began the kind of the home turf with psychology journals, but the scope has been broadening. We need more medical journals. There's 1, maybe 2 medical journals that are doing it now, but would really like to see that break into the big name medical journals. And I think that would really create a sea change. So here's what I would like to see more of. I'll let you read the caption there. And this is just life. Sometimes studies not every study has to turn out positive. There will be some negative studies. So are these efforts making a difference? Just very short on time here, so I'm going to get to my update article in PLOS Medicine in about a year ago, January of last year, with some colleagues, and we found again the old 50-50 split between positive studies and negative studies. This is only 4 drugs as contrasted with the 12 drugs we did before. But interesting, we still got the 50-50 split. But what you see here, refreshingly, is that these white blocks here these represent negative studies where they're not significant on the primary outcome, to them being negative. Shocking. That just hardly happened at all in the previous data set. So here are some analyses. This is the older cohort where you had the p-value based upon the journal, and on the x-axis p-value according to the FDA. And you see there's a bit of scatter there, of course, in the older cohort. But with the newer cohort of studies, they line up much better. There are still some unpublished studies, and here's a case of outcome reporting bias. But I think this looks better than the right panel looks better than the left panel. And this graphic here shows the proportion of positive. I want to emphasize the green line at the bottom. These are the subset of negative trials, and how the proportion transparently reported has increased from 11% up to 47%. So the glass we still have a long ways to go before that glass is full, but at least it's moving in the right direction. I'm going to have to, this is an effect size look. There's more concordance between the FDA-based effect size and the journal-based effect size. And just to wrap up in the next couple of slides here, this is a slightly off topic. I've talked mainly about more standard types of publication or reporting bias. There are other biases that we need to watch out for too, such as hot stuff bias and spin bias. So I'm going to show you, this is the catalog of bias put together by the Center for Evidence-Based Medicine at Oxford. And so here's an example of both of these combined. Here's hot stuff, people who are in mental health. You're hearing all this stuff about psychedelics and paradigm shift and everyone's totally head over heels with it. So I want to point out that it says double blind right there in the title, and also talks about it being double blind in the abstract. Yet buried deep in the discussion section, you see that at the time of study unblinding, it became apparent that at least 10% had inaccurately guessed their treatment arm. Now that's worded awkwardly, inaccurately guessed. Another way of rephrasing that is about 90% accurately guessed their treatment arm. So this was not, so there's a little bit of spin there. And so it's spin in two ways, the way this is worded here, and the fact that it was buried in the discussion, and that probably 95% of readers are going to see the double blind in the title and the abstract and not get to this or even think about it. So this is, I'll leave you with this here. This is a cartoon. This came out a few days ago. I saw it on Twitter, and this is are these really double-blind studies? So final wrap-up slide. Conclusions: reporting bias can skew the risk-benefit ratio. It's not just psychiatry. It may be a number of drugs. Perhaps guilty till proven innocent. Maybe we shouldn't just blindly accept everything we see in the peer-reviewed literature. There are two main types: the study publication bias and outcome switching. It seems to be baked into the peer review system. Transparency efforts do seem to be helping, and we could use some less focus on success and wishful thinking and more focus on the truth, even if it's boring. We do need to still be vigilant, and we need structural change in the peer review process. And that is it. Thank you very much. I'm sorry to run late.
And thank you, Eric. That was fantastic. And don't worry about being late. We probably won't get to all the questions. There's a ton of them. So you've obviously triggered some people to think. But I'll start. If you want to have a quick sip of water, I'll just do some housekeeping. Remember that if you want continuing education credits, please complete the evaluation form and submit it as soon as you can. And we'll get the credits out to you shortly. And if you're a prescriber in BC and you haven't signed up for our prescribing portrait program, go to our website and check it out and see if it's of interest to you. And if you want to talk to us about it, get in touch with us. So Eric, I'll start with and get through.
Is it possible if people have questions, since the time is so short, could they email them to you or something, and then you could forward it?
Oh yeah, for sure. We'll keep a record of the chat, and then I'll send them to you, and then we can maybe post the answers on the website.
Excellent. Thank you.
So the first one was there a response from Big Pharma to your original New England Journal of Medicine study about the difference between the FDA and journal publications?
They largely kept quiet, hoping it would blow over, but it got too much attention in the press. And so, yeah, there were a couple of, one company wrote a letter to the editor saying that, oh, you're making too big of a deal of these negative studies. They really aren't important because they're not interpretable. Anyway, don't want to get into the weeds about that, but they tried to argue that these should have been unpublished. The ones that were unpublished should have been unpublished. And we responded, look, you should publish it. Let the community decide whether they should or should not have been published, but at least be transparent and
Sure.
Tell people about it.
Okay. Do journals get reprimanded at all for publication bias?
Sometimes they do. Yeah, there was, I'm thinking of Ben Goldacre in the UK did there's probably many other examples, but did a compare project where he created a website where he compared something, what the study outcome was in clinicaltrials.gov with what the journal said. And anyway, he posted it, and he got some, I think Annals of Internal Medicine was one that particularly kind of went, and
Interesting. And here's I think a fascinating question. How do we talk to prescribers who have great faith in the efficacy of antidepressants, given what you've presented?
Well, I'm a psychiatrist myself, and I do prescribe antidepressants, and some people might be horrified at that. What I've presented is that they're less effective than it would appear from peer-reviewed journal articles. I try to just sort of be realistic with patients when I present them. As sometimes say these work. There's some people for whom these work great, but there's other people that we might get a partial response, and there's some people that get no response, and we can try a second or a third, so forth, and need to be patient. So I'd rather they have realistic expectations up front rather than get their hopes up falsely and and then they just give up after the first one that doesn't pan out as well as they like.
Sure, sure. On a related note, how many people, when you are realistic or set realistic expectations, choose not to take antidepressants?
Well, there's some selection bias in there already because they've already been sent. There's been a lot of prep time, and perhaps perhaps they've been working in psychotherapy for a while, and they've gotten what they can out of that, and there's really nothing further for them to do except to try medications. And maybe they've already been fighting the idea of, oh, I don't want to take a medication, and so they finally, well, maybe I should give it a try. So I don't know.
Sure. So this comment, many modern psych drug trials involve taking subjects off their current meds. How many placebo subjects are actually people in drug withdrawal? And has anyone tried to adjust for worse outcomes in placebo subjects for this reason?
If they were randomized to placebo from the beginning, then if they get taken off placebo, of course, they're not coming off of an antidepressant. But there are some trials where there's a short-term trial, and I'm thinking of esketamine, the example that we gave of esketamine, a drug that we mentioned earlier. There was a maintenance trial where patients who did well on esketamine were then randomized to either continuing on it or to placebo. And I think withdrawal was it had to be happening there. Plus, not only, and even if there wasn't withdrawal, it had to be way obvious to people that they were no longer on this drug, because esketamine is an anesthetic. And if you were taking something that makes you potentially fall asleep or have depersonalization or other weird side effects, and then you suddenly stop having them, then the blind is broken. But yes, there's other issues with just standard antidepressant trials when a person comes to the end of a trial and the trial's over. And I think the FDA has historically given very little attention to what happens once a clinical trial is over.
Okay, and it's 1 o'clock, so maybe I'll just ask one last question, and then if you have time to get to them at a later date, the rest of them. What do journals require from submitting authors with respect to disclosure of the full extent of their statistical methods? And are there uniform published criteria that outline disclosure standards?
If I understand the question, I believe that's going to depend upon the journal, and some, if they insist on a protocol, and some might potentially insist on a detailed version of that, a statistical analytic plan. But I would imagine there's quite a bit of variability of that, and they generally, probably most, don't ask for that would be up to the reviewer. And some journals have a statistician that they have review, and others do not.
Great, thanks. Welcome, everyone. I've got Dr. Erick Turner with me, staff psychiatrist at the Oregon Health Sciences University in Portland, Oregon. And today, we're going to go through some of the questions and answers that arose from his webinar that he gave a couple of days ago in the middle of February for the Therapeutics Initiative Best Evidence webinar series. So Erick, thank you so much for agreeing to respond to some of the questions we didn't have time to get to during the webinar. And the plan is I'm just going to convey the questions or comments to you and give you a chance to respond. How does that sound?
Sounds great. Thank you.
Okay, so Erick, first question. Was there a response from Big Pharma to the New England Journal of Medicine study trying to justify the difference between the FDA evaluation and journal publications based on your original paper from 2008?
Yeah, there were a couple of comments. One was in the form of a letter to the editor by Wyeth Pharmaceuticals, who makes venlafaxine. And then another was in the form of a letter to the editor of the New York Times by Eli Lilly. And I can try to briefly talk about both of those. In the first case, the letter to the editor was arguing that we should not be making a big deal about unpublished trials that are negative because negative trials really don't mean anything, shouldn't be, are uninterpretable and not scientifically valuable. And so it's just as well that we forget about them and not make a big deal of them. So our counter argument to that was that it's a bit, well, not so many words, is a bit patronizing of you to you, the drug company, to be deciding for us what should be considered valuable and not or not. And perhaps you should go ahead and publish it and let the academic community decide for itself as to whether it's truly scientifically uninterpretable and not worthy of their time.
Sure.
So,
oh, sorry. Go ahead.
Oh, and the other case was the New York Times, a letter to the editor, and they were saying that a couple of trials of duloxetine were in fact published that we showed as being not published, but in fact, what as you look at those trials, their definition of publication, the devil's in the details, and technically you could say they were published, but they were published in the form of review articles. They were bundled, and I think I touched on that topic in the talk, the pooled publications bias. You take a positive study, it gets its own whole journal article devoted to itself, but if a study is negative, well, it can be tucked into a review article along with a half dozen other trials perhaps, and it becomes less noticeable.
Yeah, I've used that technique at home when I'm supposed to go grocery shopping, and I secretly, quietly mention that I stopped at the liquor store and restocked the beer fridge. So I can see that happening totally. But it's interesting. You and I have talked about this on Twitter before, about this concept of positive trials and negative trials. And really, when you think back to the scientific method, no experiment is positive or negative. It's all informative, and it's a, you know, having all of that available, like you said, let the academic community decide. It's the only way to see the big picture and get to the truth. So I totally agree with your sentiments.
And the truth is not all right. I think we're accustomed to thinking the truth is should be positive. It's somehow that there's a what's the word I'm looking for? You know, a bias, if you will, against negative trials, that somehow the investigator has failed or the drug, you know, it's a failure of some sort. But yeah, it's not every study is destined to be positive, and even things that work. Overall, when you meta-analyze them, there are some negative studies in there. It doesn't mean you don't have to have 100% batting average with every single study be positive.
Exactly. That's a fantastic analogy. I might steal that for presentations, the batting analogy.
Yeah.
Thanks, Eric. The next question, what about trials that weren't reported to the FDA? Presumably the reporting bias with that subset is even more disappointing. Now, has anyone done an analysis, or your group done an analysis on that?
Well, the question is, see, that trials not reported to the FDA in the trial, and I think I touched on this point during the talk, but it was very, very brief, so it could have been easily missed. For a drug company to do a trial, at least in the United States, before they can even start the trial, they have to tell the FDA about it. Now I'm talking about a pre-marketing study. I'm not talking about a post-marketing study after something's already on the market, but something before the drug has been brought to market. So the FDA has been informed by the drug company that the study is going to take place, and then they go do the study, and then it might turn out negative. And then after the results come in, and they send their new drug application, their sort of dossier of all the studies that have been done. If they try to omit certain studies, the FDA is going to say, what are you trying to pull here? You know, you told us that you were going to do, you know, you said you were going to do studies A, B, C, and D, and E, and you only send us B and C. What's going on?
Yeah.
Yeah. So now it could, the loophole would, I mentioned, yeah, these are studies in the US, so it's possible that studies could have been done overseas, prior to the drug company ever approaching the FDA about about that it even wants to them to consider a drug application. So,
and that insight into the regulatory process is fascinating, and I'm not sure if people are aware that there are these giant loopholes. So just because you get licensed for sale in the US or Canada, that to assume that the regulators had access to all possible information is false. I remember a colleague of ours, Dr. Minsis, did an analysis of vortioxetine, and in that paper, in the discussion, outlined that in some cases, regulatory agencies, I think the FDA, you can correct me if I'm wrong, but do not require a full systematic review of evidence. The new drug applications have some specifications about what's to be included, but it's not necessarily every single trial that was conducted for that particular drug.
well, I don't know. I think, well, one thing I wanted to add is sometimes the regulatory agencies have have sort of back channel communications, and they can compare notes sometimes and see if something's being omitted that way. But the concept of systematic review, I worry about that. It's a systematic review is basically a manuscript-based approach, so using the published literature, and the it's sort of held up on a pedestal as being the pinnacle of at the pyramid of evidence-based medicine. Systematic reviews and meta-analyses are considered the pinnacle. But the problem with systematic reviews is that they rely entirely on what's published. They begin and end. Usually, they seldom look for unpublished evidence. Mm-hmm. And the idea is if you systematically search the literature, that, well, you've covered everything. Well, you haven't covered everything because you missed the unpublished studies.
Got it.
And I would like to see a pyramid or some other sort of model of evidence-based medicine that takes into account the question of non-publication and spin outcome reporting bias as well. So I'm not too sure I fault regulatory agencies for not doing a systematic review, because I don't think they're going to learn anything by looking at the published literature. Again, they've been informed of the study's existence before the study even began, and at which point everyone is optimistic that it's going to turn out positive.
Yes, absolutely. And our group at the TI have had our own experiences with our conclusions of a review with published literature only, and then going to some of the information that we found on the US FDA site after learning from you years ago of how to mine that site for information, and everything changes after that. So it's a good point. The next question, it's actually more of a comment. I think they're referring to your initial slides on your 2008 paper. I saw similar differences between journals and FDA with drugs other than antidepressants and outside the psychotropic drug classes. For example, drug safety studies in other areas. So I think you had mentioned in your slides that narrative review article on reporting bias, that this is not unique to psychiatry. It's found in all aspects of clinical work.
Yeah, I think that I'm glad the person made that point, and I think that's not is overlooked. I think with the one of the outcomes from the work that I've done is that because I used antidepressants in this study, and then went on to look at some other drug classes within psychiatry, one might there's the danger of a term I learned at the FDA, pseudo-specificity, that one might, this pertains only to antidepressants, or only pertains to psychiatry. It just means we haven't looked in other areas. But indeed, and I think that before we beat up too much on antidepressants or psych drugs in general, we shouldn't let other drug classes off the hook until we've taken a similar level of scrutiny with them, which still needs to be done. But yeah, there has been some, but not quite as much as the psychotropic drugs have gotten.
Yeah, you might say it's not the drug class, it's the humans that cause the problems.
Right.
with regard to reporting bias. And I think not emphasizing the negative aspects of your work and only the positive is just part of the human condition. So ignoring it is a mistake. So there's a comment here about does Erick have any comments on this BMJ study published last year from Mark Stone, in which they conclude response to acute monotherapy for major depressive disorder in RCT submitted to the US FDA. It's an individual participant data analysis. Now, I'll pull it up and move on to the next question, and then maybe we can talk about that. So here's an interesting question. How do we talk to prescribers who have great faith in the efficacy of antidepressants, etc., when there is information like you've shared, the work that you've done? So do you have any suggestions for clinicians that are maybe dealing with other colleagues that are more positive about the possible effects of antidepressants, or specialists for that matter?
Yeah, here, I guess, here's the focus is on antidepressants as if they're the only class of drugs that has efficacy issues. I think I don't want to let other drug classes off the hook just because we haven't looked at them with the same level of scrutiny, just as we mentioned a few minutes ago. I do try to emphasize to patients to whom I prescribe that these are not perfect drugs by any stretch, and it's a bit hit or miss. There are some people who get very fortunate and have a wonderful response, and but other people who have sort of been around the block and tried a number of meds and they don't work. And so if they've tried 4 or 5 antidepressants and they haven't worked, then number 6 is highly unlikely to be a home run hitter, to use another baseball metaphor. Sure. So I try to set expectations early on that basically don't get your hopes up too much. Perhaps a little bit more optimistic with someone who's having their first ever trial and hasn't been around the block, so to speak. So I don't know if that answers the question.
Yeah, totally answers the question. And I guess it also relates to the prescriber's expectations too. If you understand the evidence and you see patients come back and they don't report a meaningful improvement, then it shouldn't be a surprise, right? That's
right.
part of the expectation.
Yeah. You shouldn't be shocked and you say, "Oh, well, you are highly unusual,
yes,
Mr. Patient. I don't know what's wrong with you," basically. And in fact, I think there's the term treatment-resistant depression, which, as you think about it, kind of blames the patient in a way for not responding, when in fact it may be the drug's fault for not being effective enough.
Yeah, that's interesting that you say that, because the choice of language that's made by clinicians to their patients, I think, has a huge impact on maybe inappropriately setting expectations. It comes up when I talk to students about diabetes treatment, type 2 diabetes treatment, when they refer to someone being poorly controlled. And I think, okay, if I were the patient that you said I was poorly controlled, 1, I feel bad. And 2, what are they actually talking about? They're probably talking about my glycemic indices are not at a target. Now, in certain cases, the targets aren't justified by the evidence. So who have you helped? You're kind of blaming the patient for not getting to a target that isn't known to be helpful in the first place. So I think a careful choice of language is probably a better idea. Very interesting. And going back to that study that someone had mentioned, it's a paper published by Dr. Mark Stone and colleagues in the BMJ last year in August. And I'll just read the conclusions because I think it relates to what we just spoke about. It said the trimodal response distribution suggests that about 15% of participants have a substantial antidepressant effect beyond a placebo effect, highlighting the need for predictors of meaningful responses to specific drug treatment. So I guess the plain language summary would be about 1 in 6 to 7 people will report a meaningful effect of antidepressants. And does that align with how you would portray them?
Actually aligns surprisingly well, because if you to go back to this, we talked about the meta-analysis in the body of the talk, and we got an effect size. We didn't tried not to get into the weeds as to how effect size is, what it is exactly, but it's we had a number of 0.31 for antidepressants, and some people have a hard time. Well, what does that mean? Well, to try to just briefly summarize, it's the difference between drug and placebo in terms of number of standard deviations, although then we could get into the weeds about that, but it's still kind of amorphous and a bit difficult to understand. So people say, well, can you make that a bit more, can you put that in other terms? And one other measure of effect size is the number needed to treat. And it turns out you can actually, there's a computation where you can convert the effect size of the standardized mean difference of 0.31 into a number needed to treat. Number needed to treat, how many patients you have to treat to get one response, which is a little problematic in my mind because it dichotomizes, either a person responds or they don't respond, but let's leave that aside.
Sure.
But anyway, it turns out that 0.31 translates to number needed to treat of 6, which is exactly what you said.
Yeah. Okay. Yeah, it's interesting. And who out of, if you have a patient in front of you, I guess the discussion would be, I can't tell if you're going to be that 1 out of the 6 people that I've recommended, so. And
it doesn't mean the other 5 have 0 response or a negative
Absolutely.
response. Yeah. It's not like antidepressant response is not like a light switch. It's either all on or on off. There's all these shades of gray in between.
Interesting. Yeah, that's also important to add. Okay. Let's see. Many modern psychiatry drug trials involve taking the subjects off their current medications. How many placebo subjects are actually people in drug withdrawal? And has anyone tried to adjust for worse outcomes in the placebo subjects for this reason?
I wish I had a good answer for that. That's I think we'd have to look at the protocols of some recent studies to see if they screen for that. If I can't recall one way or the other. It's been a while since I was at the FDA and looking at those protocols. I don't recall seeing. I just don't have a recollection on that.
Okay.
Have to shrug my shoulders on that one.
Okay, no problem. One comment from someone in British Columbia said, you were talking about, I think the term is harking, where you keep on doing tests or you do a number of tests hoping that you can go back and then find the one that was, is that the correct term, harking?
Yeah, there's a number of terms. There's harking, hypothesizing after the results are known. There was p-hacking as another one.
I think it's p-hacking that you're talking about. And related to this comment, so she said she experienced working with one PI, principal investigator on a clinical trial, that ran 17 different analyses in order to find significance in hopefully one. So it happens.
and I've heard that from, there's a resident at OHSU that I was on call with recently, and she said the same thing, that had been her experience. And in med school, she was doing research with someone, and she had been really excited about research, but then when she kind of saw that, she thought, this is not right. And I personally had that experience as well when I was at NIH. I think we considered it a game to sort of see how, it was like a badge of honor, how smart we were, that we could try we knew all these statistical tests, all these options that we could try. Aha, look, hey, I found something here. And we didn't think that it was anything wrong with that at the time. It just seemed that was the way that the game was played, and that's what the big boys do. And I think I touched on this concept of social proof, which is sort of like, well, this is the way other people do it. It must be okay.
Yeah, it's scary. So on a related note, this is a question for me. Is there evidence that journals hark? And I have a personal anecdote, and it was a small, tiny experiment done with pharmacists trying to intervene and get rid of drugs that didn't have an indication. And the small experiment determined that the impact of pharmacists in that particular area didn't work. And so we tried to publish that. And in the discussion, I remember saying things like, there are potentially reasons that it didn't work because we only gave the message to prescribers one time, but in the end, we didn't have a meaningful improvement, or it didn't lead to meaningful improvement. And the journal said, no, we're not interested in that. You didn't find anything. So
Okay.
has someone done an analysis of journals and their refusal of studies that don't show positive effects or anything related to harking or p-hacking?
That would be a wonderful study. I think, I mean, you know what's happening, and what you just described, the outright rejection of the manuscript, I think points up the need for the registered reports approach, which I mentioned towards the end of my talk. So I bet if you had written up the rationale for the rationale for your study and the methods, and then let them evaluate that without the results, they might have said, this is a very interesting question. We think it's important and it should be answered one way or the other.
Mm-hmm.
And then maybe it would give you some feedback as to maybe you should tweak your methods in this way or that way, but it wouldn't be based upon the results. And then let's say that they say, yes, we think this is good science. And then you go do your study and it turns out negative. Then they can't turn around and say, oh, actually, this isn't so important after all.
Yes. Yeah, it's the registered reports thing would be an elegant solution to this problem. So hopefully it keeps its momentum going. I remember,
if I can pick up on the registered reports thing one more time, and my attraction for that is I see similarities with the FDA approach, because again, the FDA learns of these studies before they're started, and they then get the protocol, and that they have the opportunity to give feedback to the drug company before they start the study. And then crap happens down the road. Some of the studies don't turn out well. And then the drug companies will sometimes, oh, can we use this scale instead of that scale? No. You said that you were going to use the Hamilton Depression Rating Scale and not the MADRS. I'm giving you a real example where they said that the HAM-D was going to be the primary outcome, and the MADRS, this other depression rating scale, was going to be one of the secondary outcomes. And guess what? The MADRS was positive, the HAM-D was negative, non-significant. And they said, well, can you actually, the HAM-D is not really a good measure. Can we use them? No. Next study you can.
Yeah.
But don't try to change if it turns out the other way around. Don't try to change that on us again.
Yeah.
You have to live by the contract up front.
Exactly. Yeah. But people try. I remember reading, we had done some survey work of journals about, do they have a method to detect outcome reporting bias, like the big high impact medicine journals. And what we found was, it was interesting because the journals, before answering our questions, asked for anonymity. And if we wouldn't do that, they wouldn't answer our questions. But what they said was interesting, like, we don't routinely check for reporting bias. Like, we don't, if someone wants to submit a paper to us, we don't always go look at the protocol to make sure that they did. And because we have other priorities, it takes too much time. Well, then who's supposed to do it? And they said, well, and some of the journals said, the peer reviewers should be doing it. And then we asked them later, do you think the peer reviewers are capable or know that that's what they should be doing? And they said no. So it's just very strange to me that there's no, it'd be a relatively easy system if there's a published protocol or a registered protocol that you could quickly check. The other thing that I read a paper that came out after ours that talked about, talking to the editorial groups of the big journals and asking them why this goes on. And I can't remember the citation, but one of the comments from a journal was, if we don't publish a paper where we've detected that they've changed the outcomes that they're going to report, and we hold our standards high, that person, and we reject that paper, that group is going to take it to the next journal and they'll publish it. So, we have an interest in getting this out there. So sometimes I think the message or the implication was sometimes we overlook those discrepancies. But it's troubling.
That's kind of like, I don't know, reminds me of some political thing about if we, should a certain U.S. president and, people that work for him and say, well, why didn't you leave? Oh, well, someone needed to be the adult in the room and I made it better by staying.
Yeah.
Like a harm reduction, I think is the phrase.
Okay, let's see.
It'd be worse if they took it to that other journal.
Yeah, exactly. Yeah, there's always justification. Someone commented on partway through your presentation, this is all starting to make me feel a little depressed. Has anyone tried to explicitly evaluate how powerful the placebo effect can be when treating mental illness? Like, for example, comparing two different placebo arms? Or can you comment in general on the placebo response for major depressive disorder?
That is a big topic. There have been people that have looked at that. Irving Kirsch comes to mind, who's looked at antidepressants also and used FDA reviews, and he and then some others have looked at the baseline severity of depression and how that relates to the likelihood of how much separation you get between the drug and placebo. And basically, starting out with more severely depressed patients, and you're going to have less placebo response and more antidepressant true response. And others have looked at that since. And something tells me that someone has found an opposite, a finding contrary to that. But most of them have found what I just mentioned with the severity, baseline severity. But you can kind of see how much placebo response there is in a typical clinical trial. You can see the curves come down from baseline, and I don't have one in this PowerPoint, but both curves will come swooping down, and then the study ends at, say, week 8 typically. And so most of the decrease is occurring in the placebo group as well, and you see some separation. So on average, there's very rough numbers, maybe using that Hamilton Depression Rating Scale, if that's the primary rating scale, you might get a 10-point decrease in the active group and an 8-point decrease in the placebo group. So you could say, well, 8/10 or 80% of the response was, to use a phrase from Irving Kirsch, duplicated by a placebo. Now, does that mean, and then we can get into a whole thing about, why is that coming down so much? There's, I don't know if you want to talk about that, regression to the mean is one of them. All the attention that people are getting in a clinical trial, being, interviewed every week and thinking, wow, I'm really doing, this is really exciting what I'm taking part in here. Anyway, but some people have really devoted their studies and careers to looking at placebo response. It's potentially a big topic.
Yeah. And, those curves you talked about, Tom Perry and I and others in the TI had looked at non-opiate drugs for chronic pain. And you see the same thing. If the study reports pain scores daily or weekly, within the first week, all the scores drop, regardless of treatment assignment. And then the separation occurs after the first week. But, compared to the big drop, it's marginal after that. Yeah. So the same phenomenon. It's interesting. Not all the trials like to look at the data that way.
Some people have looked at like big pills versus small pills, and injections versus oral, and a lot of these things make a difference.
They do. Okay. the next comment or question, there have been at this point numerous meta-analyses of SSRI efficacy relative to placebo, and all have been very consistent in showing a fairly small effect size and high risk of bias in both studies. What differs among these meta-analyses is the interpretation, such as the clinical significance of a 2-point difference on a 54-point Hamilton Depression Rating Scale. Can you share your thoughts on this?
Yeah, I definitely agree with the right. The results themselves are pretty consistent with one another, and then how they're interpreted, they can be spun one way or the other depending upon, you can say, well, obviously this is a trivial difference, and others might say, oh, this is a huge difference. This is very important. But these are opinions. So I think it's hard to I don't know. I guess I've sidestepped the question of clinical significance, but because it's hard to it's not I mean, it's one thing to measure the effect size, and it's another thing to kind of come down from on high and say this number is below a meaningful threshold, and I hereby declare this to be clinically insignificant. It's a little bit patronizing to say that when we don't. Because clinical significance is going to differ depending upon the individual, for one thing, but it's going to depend largely on the indication in question. If you're trying to treat cancer, for instance, sometimes the differential between active treatment and and existing treatment, the new, if you add on and say a new treatment to existing treatment, sometimes they're trivial differences, and it would be kind of a hot topic to be saying, okay, we're not going to give you chemotherapy if you're a cancer because in my I am the authority and I deem this to be a insignificant difference. It's I don't know. It's a really tricky thing. On the other hand, we could get into talk about speaking of oncologic drugs. Some of them are so horribly expensive, and do we pay any price for something whose difference is going to extend life by very little when the outcome is almost certainly going to be the same.
Exactly. And I guess
very
it depends
tough questions.
Yeah, very tough questions. And it depends on, are you a public healthcare funder or the individual sitting in front of you whose family members suffer? Those perspectives change the interpretation. And I guess this idea of clinical significance, listening to you talk, it's, did the effect in the meta-analysis cross that threshold is almost irrelevant when you're considering the patient sitting in front of you, because if the effect size in a study says that the average effect is not clinically significant,
right?
But you're treating me, and I report back to you that my life has changed. Some aspect of my depression is gone, and now I can function. Then it would be ridiculous for you to say to me, Aaron, that's not possible. You must be making it up, because the paper I just read said there's no clinically important effect, right? So from the point of patient care, it might not make a difference.
Yeah, the point, inter-individual variability, and conjures up for me the old joke about the statistician with one foot in boiling oil and the other foot in liquid nitrogen. He says, on average, I feel just about right. So we've got these averages that we look at in these studies, but it doesn't tell you about the individual patient. And you may have someone that your next patient may be someone that could have a good response. And also the person's preferences. That's a big part of evidence-based medicine. You can't just sort of use EBM isn't intended to be a bludgeon, or a club that you hit people over the head with and say, one size fits all. You've got the people's preferences and their situation. If a person say, hey, I've tried everything, I've tried therapy, I've tried exercise, and nothing is working, can you please, I'm having suicidal thoughts. Do you just say, tough?
Yeah.
No, I'm not
Yeah.
going to treat you.
Yeah. Yeah. It's fascinating. And one thing I've tried to figure out is it's different when you're using drugs to prevent complications in the future, like cardiovascular drugs to prevent a heart attack. You never know in the individual if it's having the effect that you desire, right? If they
Right.
come back and have a heart attack, doesn't mean the drug, like it's impossible to say that the drug failed. So anyways,
Right.
but with the treatment of symptoms or signs like depression or pain or anxiety, I think the best evidence is how I tell you the drug made me feel after I took it, regardless of what the studies say. Like if the studies say overly the drug is going to work, and I tell you I tried it, it doesn't work, you're not going to tell me I'm dumb or I
Right.
misinterpreted,
Right.
right?
Right.
So, um,
Now, the flip side of that is if
yeah,
someone comes in and says they want to go on something for which there's no evidence or very little, just anecdotal evidence, they want to try some, what was it that a certain political figure was talking about using Clorox or
oh
something,
yes,
or
yes,
ultraviolet light and internal organs, and some patients say, I want to do this, and you say, I don't think that would be
sure,
wise. But who are you? How dare you tell
exactly,
me? I get to. So anyway, I think evidence, the average evidence is certainly good, is the best starting point, and
yes,
then but that has to be refined based upon feedback from the patient and how they respond.
yeah, exactly. Okay, on it, I could talk to you for hours, Eric. This is fun.
Yeah,
Um,
absolutely.
okay, would there be a benefit to pushing for legislation for companies and universities to publish their negative data if the trial is registered? I'm aware that there would be huge obstacles to overcome, like lobbying, partisanship, etc. But do you have any comments on that?
Well, we kind of have we have a law already in the U.S. about that they have to post their results in clinicaltrials.gov. So that's a law now. Has the law worked? Well, yes and no. There's been a lot of quite a bit of non-compliance, or the more politically correct term, non-adherence to that to that law. As far as publishing in journals, that is that would be taking it to another level, and you could make an argument for that being necessary because I think most clinicians aren't really tuned into the idea of looking at clinicaltrials.gov for results of studies. I think they are accustomed doing literature searches, if at all, or just hearing about a certain paper through the news or word of mouth from colleagues and things being in journal articles.
Mm-hmm.
So,
Agree. And
yeah, we do have the legislation, but it doesn't extend to journal. And then you have the right. Well, what about who are you? How would the journals feel about being told you've got to publish? I think maybe they should have to publish,
mhm,
negative studies as well, and not
mhm.
be able to pick and choose based upon the most impactful, what they deem was going to get them the most, eyeballs and hits and news reports.
Yeah, if the journals didn't have profit associated with their business plans, then it might be an easier sell, but that's not the
Mm-hmm.
case at this point. Uh, what do journals require from submitting authors with respect to disclosure of the full extent of their statistical methods? Are there uniform published criteria that outline disclosure standards?
Not that I know of. I've heard of some journals requesting, more recently requesting protocols, but I can't tell you how many journals that I think that would be a good study to do. And maybe this has been done and I'm just not aware of it, a survey of journals and see how many require the protocol up front. And then, and then how detailed the protocol has to be, if the statistical methods are
Yeah.
fleshed out enough so that a person can replicate the same results using the same data set and using their methods in the protocol.
Yeah. In our small evaluation of the medicine journals, we asked them, do they always check the protocols? And they said no. The vast majority of the few journals that we talked to said we don't always double check the published or registered protocols to make sure the outcomes match.
Yeah, send that to me if you would.
Sure.
That'd be great.
Yeah.
Yeah, and then it would be easy for them to get the
Absolutely.
protocols and just look at see what they're emphasizing in the paper, and then compare it with the protocol. And if it's a mismatch, I remember looking at reviewing something years ago for I think it was PLOS clinical trials, which later became PLOS One, and a manuscript came my way, but I had access to the protocol, and there was a mismatch. It was the intent of the study was entirely different, and I didn't want to look at it any further. That was it, as far as I was concerned. I mean, if they're going to spin things around to do a study and try to obviously make put heavy amounts of lipstick on a pig, then I'm not interested in it. And it's dishonest.
It is dishonest. And with AI and journal submission websites, you could just have them populate fields in the submission process. What are the outcomes at what time point, whatever. And that, just fill the database. So it potentially could be quite easily done with technology. And then the cross-checking would highlight red flags, and then you would go back. Anyways, maybe we can build a system and patent it and make tons of money, Eric.
There you go.
Okay. I see.
But we'll only publish the positive studies about it, though.
Of course. Okay, I'm going to have to speed up because I'm talking too much. It's too interesting talking to you. What about reporting? What about reporting bias for safety outcomes? Are you aware of that specific area being analyzed? Or
yeah, I have not looked at that much myself, and I think that is a hotter topic. I mean, and by hotter I mean inflammatory. I think when you find things that are being deep sixed about safety signals and problems and deaths and stuff like that, then I think I know it. I know it has been done. One of the problems with looking at safety is it's more difficult than efficacy. You could take these study results and just combine them into an effect size and get a standardized. You get one number from every study. With safety problems, you got maybe a problem with the kidney and a problem with the liver and maybe suicidal events, and they're all qualitatively different, and it's difficult to kind of put them together or kind of wrap your mind around around it. You usually have to pick on one thing or one domain to look at.
Yeah, it's much more diverse in the kind of information you're dealing with. I think, is it Sonal Singh and colleagues? I've read reports where they analyze the adverse event reporting based on unpublished data and published data, and you're right, it's as bad or worse. Okay, how about is there going
you'll
to be...
see, actually, you'll find, sorry to butt in, in the interest of, I'm not helping you out as far as the time goes, but
that's okay.
I mean, there have been lawsuits. I mean, every now and then you hear about lawsuits where a drug company has swept a certain safety problem under the rug, and then it comes out, and so we know it happens. There's no question that it happens. Then drugs get pulled off the market. Vioxx comes to mind, but there are many other examples. So
exactly,
sorry.
there's one other one. I think it was related to dabigatran, an oral anticoagulant, and what they knew about the ability to assess the level of anticoagulation, but kept secret because their marketing plan was to stand out from warfarin, which requires it. And lots of tasty examples. Unfortunate. So here's a question. You said that there's a law in the States about registering protocols in clinicaltrials.gov. Is there meaningful enforcement that you're aware of?
No.
Okay.
No, there is not. In fact, there's a whole database online. I'll give it a plug. FDA. Okay, the name of the law is FDA, FDAAA, Food and Drug Administration Amendments Act, and the website is Trials Tracker, and they see how many of these trials are getting reported, and how many have results reporting, how many don't, and trying to think, guy's name slips my mind at the moment, but yeah, there's been very poor compliance with that.
Mm-hmm.
And there's supposed to be fines associated with that, and some, there's one website that's talked about called Transparamed, who's looked at the millions, perhaps billions of dollars that have been left on the table, where the FDA could be and should be fining the drug companies for not putting their results in clinicaltrials.gov. And the problem is, why isn't the FDA enforcing that? So that's been, people have been lobbying the FDA. What's, why you're not following up on this?
Sure. So Eric, maybe I'll give you the last word. Where do you see, what do you think is the next step, or what do you think clinicians or policymakers or academics, investigators, what could we do tomorrow to help fix this problem, or start to fix this problem?
Well, yeah, don't revere the peer-reviewed literature. There's nothing about the peer review process that renders something the truth necessarily. So let's not be naive about it. It's we're kind of on the lookout in other domains, like when, at least in the US, we got our commercials on TV, and when someone puts an ad on there, we say, okay, well, yeah, they would say that they want to sell us this drug or this soap or whatever it may be. So peer-reviewed articles go through a more rigorous process than that, because they're reviewers, but things can still slip through, and they do slip through, and because of these problems. So I think we can't be nihilistic. On the other hand, we can't disbelieve everything. We just have to be, skeptical, but educated but skeptical, and not naive, if that's perhaps redundant there. And the existence of publication bias, I'd say, does not mean that we should throw the baby out with the bathwater. To hell with it, nothing works, and we should just give up. I think it's baked into the system largely, because largely people are allowed. I mean, the way it's done, has been done classically, as people do their studies, crunch the numbers, get the results, and then decide whether they're going to publish it, and they're able to p-hack and whatever, and then submit that to the journal. And then you've got the journals that get to do their own, have their own hindsight bias. Oh, this was, as you personally experienced, you got a negative result, so it must not have been important, so we're not going to publish it. I think still register reports is to me what I'm very optimistic about. If we can just get more buy-in from journalists, especially the big name journals. There's over 300 journals that are currently using that, but so far the big fish are quite happy with their position in the pecking order. And
well, there's some good practical and there. Thank you so much, Eric, and hopefully we'll have you back again.
Be happy to. Thanks very much, Aaron.